Hello, my name is Dr. Zach. I'm a clinical geneticist, and I'm going to talk to you today in the second of 3 lectures on helping you to pass the genetics portion of the MRC PCH Part 1. And in this lecture I'm going to talk about some of the commoner genetic syndromes and how you'll answer questions correctly on those. So to do this, what I'd like to do is to give you some appreciation of the range of genetic syndromes, but also to give you an approach to how you cut this task down to manageable size. And so to hopefully give you an approach to learning about common genetic syndromes, and then to help you think about some of the features of genetic conditions, common features which might be seen regularly, which may or may not be useful in making a diagnosis, specific diagnostic criteria which are commonly used in certain genetic conditions, complications that can arise in individuals, and how we go about testing and when we don't necessarily test. One of the things that people sometimes find when they're trying to learn about genetic syndromes is they feel overwhelmed by the huge numbers of very rare conditions. There are, of course, thousands of syndromes caused by genetic mutations. The Mendelian Inheritance in Man catalogues over 10,000 conditions due to single gene mutations, and there are probably at least another 10,000 conditions due to various chromosomal rearrangements. And so it's easy to feel overwhelmed. However, what I would encourage you to do is to try and cut this task down to manageable size. One way to do it is to think about the different modes of inheritance, different mechanisms behind the genetic syndromes, and to separate conditions out according to these. Some conditions are due to chromosomal abnormalities. These can be euploidies, that is, whole, extra or missing sets of chromosomes, aneuploidies, whole, extra or missing single chromosomes, structural rearrangements, such as deletions, duplications, translocations, and all of these can be present in various levels of mosaicism. Moving down the scale from the whole chromosome, we can then have microdeletions. Now, microdeletions are, at the molecular level, rather large deletions that can be several million base pairs of DNA, but which are not observable down the light microscope. Common examples of this are Williams syndrome, Beckwith-Wiedemann syndrome. In this situation, one would use a FISH analysis to determine that there was a deletion there, because these, of course, are not observable with normal chromosome analysis using a microscope. Now, there are 2 mechanisms which often come up in exams, mechanisms of gene imprinting. The common examples of these are Prader-Willi syndrome and Angelman syndrome, and also the mechanism of trinucleotide repeats. Now, in this hierarchy, I've sort of included them between the microdeletions and the single gene mutations, but this is a little bit arbitrary, because single gene mutations can in fact be quite large rearrangements within genes. The single gene mutations can be inherited as autosomal dominant or recessive. There are, of course, co-dominant situations, the most common example being the ABO blood group, where types A and B are both dominant to O, but neither is dominant over the other. So, of course, we can have the AB blood group. And then, of course, we can have X-linked inheritance patterns, which can be usually recessive, but occasionally dominant. And then, finally, we have to remember that there are chromosomes other than the large chromosomes we see in the nucleus. There are the tiny bacterial-style chromosomes in the mitochondria, which can occasionally carry mutations that have clinical effects. And so that's one way that you can separate out these thousands and thousands of syndromes. But the other way is to think about it in a slightly more common sense approach. So yes, there is a huge iceberg of conditions, but there's a tip of the iceberg which we need to pay attention to. We need to think about the common conditions, because you're taking an exam not to become a specialist in genetics, but to demonstrate your competence as a paediatrician. So how can we think about a condition being common? Well, you can say it's simply numerically common. It has a high incidence. So one of the commonest genetic conditions which we've probably all seen is Down syndrome, with an incidence of about 1 in 650 births. However, remember that some conditions may have a high incidence in the clinic because they produce high levels of medical need, and so they come to attention perhaps more often than might be warranted by their simple numerical commonality. For your purposes, of course, there are then those conditions which are popular with examiners, and this can be the conditions where there are difficult concepts to understand. such as imprinting or trinucleotide repeats. And so from your point of view, these are common because questions come up commonly. However, one thing that you don't need to start getting into is very, very rare syndromes or very, very rare complications of commoner syndromes. Because remember, this is a generalist exam, this is not a specialist paper. When questions come up about testing for syndromes, the underlying basis of which test to use for which syndrome is, of course, the genetic mechanism that's causing it. So, of course, you don't do a chromosome analysis to look for CF, because CF is due to single point mutations usually in the CFTR gene, which are completely invisible to the light microscope, and so you'll just see normal chromosomes when you do chromosome analysis in a CF case. However, even when we're looking for single gene testing, we do have to think about some of the practicalities of what's actually involved in doing an analysis. And this will often turn on the number of mutations which are commonly found in the gene. So, for example, in achondroplasia, the vast majority of cases have one single mutation in the FGFR3 gene. And this means it's a very simple process to analyse DNA for this one single mutation in the lab. For a condition like cystic fibrosis, most carriers in the UK will carry delta F5, where perhaps 2/3 will. But there are, in the CFTR gene, over 1,500 other recognised mutations scattered right throughout the whole gene. And it's not practical in most labs to scan the entire gene for any mutation at any point in its length. And so what most laboratories do is compromise by doing panel testing. They will do a test which will look for a panel of common mutations, perhaps 25 or 30 common mutations, which will cover 90% or so of the population. In that situation, the test for carrier status is very good. It will pick up 90-odd% of the population. But because it's not practical to routinely test for the entire gamut of all mutations known as CFTR, it's necessary sometimes to send off a sample for specialist analysis at a specialist lab. For other common conditions such as NF1 or Marfan, there really aren't a lot of clustering of mutations in the population, and there's just a scatter of mutations right across these very large genes. And what this means is that although mutation analysis is possible in theory, in practice it's far too time-consuming and expensive to do routinely. It's possible if there are specific requirements for it, but it's not routinely offered. And so in these conditions, the diagnosis is usually made on clinical grounds, often with internationally agreed protocols of clinical criteria. So let's think about question 1 and do some diagnosis of a genetic syndrome. So this is an extended matching question where you have a number of possibilities down the left, and I'll give you some scenarios and pick out the most likely diagnosis. So the first scenario is a young lad who comes with short stature, web neck, some noticeable facial features, and cardiac findings. Now your initial thought might be, with the short stature and web neck, that this sounds rather familiar. But then you may think, well, hang on, the thing I'm thinking of usually occurs or has to occur in girls. So write down now what you think the diagnosis is. In this situation, the answer is Noonan syndrome. This child has the classic characteristics of Noonan syndrome, which I'm going to discuss in a little more detail later on. And the thing which might have confused you was that the initial findings suggested Turner syndrome, but of course this is a boy, and so it can't have Turner syndrome. Here in scenario B is a girl presenting with an unusual presentation for a child of hypertension. But examining her, she has a web neck and wide-spaced nipples, and she's pre-pubertal at the age of 15. We have some cardiac symptoms and signs which are of significance here, and so choose one of the diagnoses from the list and commit yourself now. This girl has Turner syndrome. She clearly hasn't yet entered puberty, she has the physical features, and she has the cardiac symptoms suggestive of a coarctation. Here's another possible case. This is a baby who presents with jaundice and is growing poorly. Again, there are features of dysmorphic face and cardiac findings, but there's also here an important clue in the chest x-ray. Commit yourself now to one of the diagnoses here. Hopefully you picked out that this is Alagille syndrome. Now moving on to question 2. This question is about the complications of a common condition, Down syndrome. Now of course you should be very familiar with Down syndrome and its features, you may be less familiar with the complications because these are things which may have not come to your attention in the clinic very often. So, have a look through this and see which of these complications you know are associated with Down syndrome. And make your assessment now. Choose which of these you think are associated with Down syndrome. Now, in this situation, B, C, D and E are all associated with Down syndrome, but A isn't. A is an example of what's called a distractor. This is an answer which looks familiar because it's a gut condition and it begins with the letter C, but is actually there to distract you away from the correct answer, which is probably celiac disease. Crohn's disease is, of course, not common in Down syndrome. Atlaxoanteal subluxations commonly do occur in individuals with Down syndrome and are commonly picked up on neck x-rays, although fortunately symptoms are rather rare. We know that a high proportion, about 40% of individuals with Down syndrome, will develop Alzheimer's disease later in life, probably due to the dosage effect of the amyloid precursor gene. And we also know that there is an increased risk of the acute leukemias, all the leukemias, myeloid and lymphoblastic. And the actual level of this risk varies with the age of the child and the leukemia you're talking about. This is a rough middle risk, about 20-odd fold. But remember, of course, that although this is a high relative risk, it produces a relatively modest absolute risk because the leukemias are very rare to start with, and so multiplying a small incidence by a moderate number only produces a relatively modest absolute risk. Now let's move on to Klinefelter syndrome. Now, which are the features of Klinefelter syndrome in this list? Commit yourself now, write it down on paper. Klinefelter is, of course, a sex chromosome aneuploidy, and there are a number of ones of these which you should know a little bit about. Klinefelter, or 47XXY, men are infertile. They have hypogonadism. There is a very mild degree of learning disability in some, and there's an increased risk of gynecomastia, which can in a small number of cases be troublesome and embarrassing. Turner syndrome is another common sex chromosome aneuploidy, which we'll discuss in a little while, and again produces infertility. A notable short stature, around 140 centimetres is the average height, or 4'7" in old money. Lymphedema is very common, and there are a number of internal complications such as horseshoe kidneys and coarctation of the aorta in about 1/10. And a number of characteristic dysmorphic features such as a short fourth metacarpal, which gives a recessed fourth knuckle, narrow fingernails, a low hairline at the back of the head, and again, very mild degrees of learning disability in some individuals. The XYY syndrome, which is sometimes people confuse for Klinefelter simply by switching around the X and Ys, so don't let that happen to you. The XYY syndrome characteristically produces a man with taller than average stature, typically an extra 7 centimetres or 3 inches of height. There is some degree of mild learning disability in a small number of individuals, but this is a syndrome which has a rather checkered history, and sometimes in the old days called the criminal karyotype, and there were all sorts of stories about it being related to sexual crimes and murder and so on, but this has now been completely disproven. This is a very mild phenotype. And also the triple X syndrome, 47XXX, which again produces most of the time perfectly normal women who are a little taller than average, sometimes have menstrual irregularities or early menarche, and again some very mild degree of learning disability in a small number. So, moving back to the question, Klinefelter syndrome men are male, despite the hypogonadism. So A is false, but B, C, D and E are all true. Right, let's move on to question 4. Now, question 4 is a clinical scenario for you to make a diagnosis. This is a young girl referred by her GP because of a heart murmur. But you notice that she is short stature, with neck webbing, and also some characteristic facial dysmorphic features. So, hopefully, when you're looking at this question, you'll be trying to distinguish between two different diagnoses. Make your choice now and write it down. This is an example of Noonan syndrome. Now, people sometimes confuse Noonan syndrome and Turner syndrome, so let's just clarify that now. In the old days, Noonan syndrome was sometimes called male Turner syndrome because, of course, males don't get Turner syndrome, that males can get Noonan syndrome, but of course so can females. Because Noonan syndrome is a dominant condition due to mutations in gene on 12q, and so males and females are equally affected. So it's a little bit of a misnomer to call it male Turner syndrome. It's a moderately common genetic condition, 1 in 2,000 births, and the features, some of which are in common with Turner syndrome, such as the short stature, the rather wide neck and chest, but there are also characteristic facial features, as you can see in this photograph here, of this young man affected by Noonan syndrome. This rather broad head and wide-spaced eyes, hypertelorism. The slits of the eyes, the palpebral fissures, are downward sloping. If you notice, the corner of the eye nearest the nose is higher than the corner of the eye nearest the temple. That's downward sloping palpebral fissures. And the eyelids are often quite heavy with bilateral ptosis. People with Noonan syndrome often do have learning difficulties. There are characteristic cardiac anomalies with both Noonan syndrome and Turner syndrome, but they are distinct. They tend to be right-sided in Noonan syndrome, pulmonary difficulties, as well as ASD, and they tend to be left-sided in Turner syndrome, mainly in the aortic valve or in the aorta itself. So remember the distinction between Noonan and Turner syndrome, and don't get confused between the two. Now moving on, we'll think about Prader-Willi syndrome. Now which of these is a recognised feature of Prader-Willi syndrome? Make your choice and write it down now. The correct answers are A, C and E. C and E are clinical features, which you'll hopefully know about. D is a distractor, because you may know that Prader-Willi children often suffer from obesity, but not in infancy. The obesity usually comes on later in life when they start eating to their first and second birthday. The first question, genomic imprinting, is one of the mechanisms behind Prader-Willi syndrome, and this is a mechanism that a lot of people find difficult to understand, and so I'll go through it a little with you now. Genomic imprinting occurs in only a small number of parts of the human genome. In the majority of genes you inherit, the 26,000 genes we all carry, the majority of them are expressed just the same whether you inherit them from your mother or your father. However, there are a few regions in the human genome where genes are switched on or switched off differently depending on whether they are of maternal or paternal origin. And this part of chromosome 15 is one of those regions. So as you can see from the slide, on the left-hand side we have the normal situation. The maternal and paternal chromosome 15s inherited express the genes in this region differently. In the maternal chromosome 15, the UBE3A gene is normally switched on, and the two genes below it are normally switched off. On the other hand, in the paternally derived chromosome, the UB3A gene is normally switched off, and the two genes below are switched on, which means the normal situation is that we have one functioning copy of each of these three genes, and one non-functioning copy. Now then, when we get a deletion in this region, it therefore matters whether the deletion occurs in the maternal or paternal chromosome. Because if the deletion occurs in the maternal chromosome, we lose the only functioning copy we have of UB3A, because the other UB3A gene is switched off, being of paternal origin. Contrariwise, if we have a deletion in this region in the paternal chromosome 15, then we lose the only functioning copies we had of Snowpen and Nectin. And so the genetic consequences of these deletions are different, because the functional consequences of them are different. And that is why the same deletion in chromosome 15, apparently, gives totally different syndromes, because it depends on which functioning gene was deleted. Now, another way for this imprinted region to go wrong is when we get uniparental disomy. Now, uniparental disomy occurs when we inherit two copies of a chromosome from one parent. Now, you might say, how can this happen? Uniparental disomy is thought to be a rescue mechanism which rescues cells that have become trisomic. That is, in this situation, if by accident there's been a non-disjunction event and two chromosome 15s have gone into one gamete, then we would end up with a zygote which was trisomic for chromosome 15, which normally would be a lethal outcome. However, there appear to be mechanisms in the cells which can recognise trisomy and remove one of the chromosomes which is there unwanted. However, the chromosome that's removed is removed randomly, and so sometimes there are two chromosomes left behind which are of the same parental origin. For most parts of the genome that doesn't matter, but for the parts of the genome which are imprinted, clearly this does matter. Because if we end up with two chromosome 15s of paternal origin, then those chromosome 15s will both have inactivated UB3A genes, which means that it's the same result functionally as though we had a deletion. Similarly, if we have two chromosome 15s from the mother, we have two functioning copies of UB3A, but we have no functioning copy of SNRPN and Nectin, which means again it's the same as having a deletion of the paternal chromosome 15. And so you can hopefully see that because of the genomic imprinting, because of the switching on and off of genes depending on which parent they come from, either microdeletions or UPD of these chromosome regions result in an imbalance of functioning genes which leads to clinical disease. Question 6. This is a question about a relatively common genetic condition, NF1. So look at this question and make your choices now. Write down your answers. Now, what do you know about NF1? Well, NF1 is inherited as an autosomal dominant trait, and it's due to mutations in the neurofibromin gene on 17q11. As I mentioned before, although we know where the gene is and we know what gene it is, in most cases it's not practical to offer mutation testing because there are no common mutations to test for. And so this is a condition where diagnosis is usually made on the basis of clinical findings. And because of this, internationally agreed diagnostic criteria have been developed, the so-called NIH criteria. Now, for a diagnosis of NF1, an individual needs to have two or more of the following features: café au lait macules, neurofibromas, freckling in the groins or axillae, optic glioma, lesion nodules in the iris, a distinctive bony malformation, or a first-degree relative. Now, a first-degree relative, of course, changes the probability that an individual has NF1. Because if an individual comes to the clinic asking about a diagnosis of NF1 from the general population, the prior risk of them having NF1 is the population incidence, around 1 in 2,000. Whereas if they have a first-degree relative, particularly a parent with NF1, then their prior risk would go up to 1 in 2. And so you can see how having a family history is of crucial importance in making the diagnosis. The other thing to, of course, remember about this question is that this is a negatively worded question. It says which of the following statements are true except. So be careful with these questions which are negatively worded, because if you look at the question and simply answer it on its face value, you might say, question part 1, inherited autosomal dominant pattern is true. So don't put true, put false, because the question is which are true except. So in this situation, all of them are true except D, which is the diagnosis is usually made by gene testing, which of course it isn't, as I've explained, because this is normally impractical. Here's another common genetic condition, Marfan syndrome. Answer the question and write down which you think are the correct answers, and then we'll move on and talk about them. In this situation, B, C and E are recognised features of Marfan syndrome. A is just simply wrong. B is probably put in as a distractor because people sometimes think of Marfan syndrome and homocystinuria together as they have similar clinical features. Individuals with homocystinuria do have learning disability, but those with Marfan do not. Marfan syndrome is another condition where mutation testing is usually impractical, and so it's diagnosed based on internationally agreed criteria which were put together in the Belgian city of Ghent, and therefore known as the Ghent criteria. Now, this is a simplified version of the Ghent criteria. There are in fact more criteria in the full version, but just for the matter of getting this on the slide, I've simplified it for you. In order to make a diagnosis of Marfan syndrome, an individual has to have major criteria in 2 body systems plus a minor criterion in a 3rd body system. I've laid out here some of the major and minor criteria in the various body systems that an individual can have, but again note at the bottom that a family history of a first-degree relative, or of course in those rare families where a mutation analysis has been done, a new mutation, is in itself a major criterion because it radically alters the prior probability of an individual having the condition. Question 8. This is a scenario of a young boy presenting with motor difficulties who's got a family history of Duchenne muscular dystrophy. So, with this presentation and this family history, there's a strong suspicion that the boy himself may be affected. Decide now which you think are the correct answers and write them down. The correct answers in this case are that a muscle biopsy is not essential, usually, to confirm the diagnosis, and that the boy's CPK level will be greatly elevated and will have been since birth. This is the family tree we're talking about. We have a boy who's been brought to medical attention with motor difficulties, and we're told that he has a maternal uncle affected by DMD. Because he has a maternal uncle affected by DMD, if the boy's diagnosis turns out to be correct, then it means that his mother must be a DMD carrier, and given the situation it seems quite likely the diagnosis will be made in this boy. So that means that the mother, in this case, must be a carrier. In this situation, the boy's sister has a 50% risk of being a carrier because her mother is likely to be a carrier. Now, we know that about 70% of boys with DMD have a detectable deletion or duplication in their dystrophin gene. And so this is a situation where we have common mutations which can be detected in some individuals. Of course, there are still 30% of individuals in which it's difficult to make a molecular diagnosis and we have to rely on clinical features. But in most individuals, in about 70% or so of individuals with DMD, it's relatively straightforward to find a dystrophin gene mutation. We of course have the other clinical features and the grossly elevated CPK levels, which are usually good diagnostic clues, which usually mean in these situations that a muscle biopsy is not necessary. About a quarter of boys with DMD will have intellectual delay, which of course is greatly elevated compared to the general population risk of learning difficulties of around 3 or 4%. Now, let's move on to another situation where a young girl is brought for the investigation of developmental delay. Now, this is a classical presentation of the condition we're interested in here. This young girl was born normally, developed normally until her 2nd year of life, at which point her development not only slowed down but began to reverse. She began to lose abilities and skills that she had acquired earlier in life, and in particular stopped walking. And when you examine her at 7, she has a hand-wringing habit or action, which is an important clue for this diagnosis. So write down now which you think is the correct diagnosis from this list. This is a classical presentation of Rett syndrome. The regression in the 2nd year of life and the hand-wringing action are classic presentations of Rett syndrome in a young girl. This condition almost exclusively affects females, for reasons that aren't entirely clear. It's an X-linked dominant condition due to mutations in the MECP2 gene, in most cases, and the vast majority of cases occur as one-off sporadic mutations with low recurrence risks. The classic presentation is that of normal development followed by regression, and the stereotypic hand-wringing motion is very characteristic of this condition. So, we've gone through some of the common genetic conditions, but of course I can't cover all of them here in the time available. So, where should you focus your attention? Now, I've said that it's possible to try and recognise some of the commoner conditions that are likely to come up in the exam. Now, of course, I can't possibly second-guess the examiners, but what I can do is make an educated guess, and this is my top 30 of conditions which I think you ought to definitely know, but I can't guarantee that other conditions couldn't come up as well. So using my hierarchy I examined earlier, in the chromosomal conditions, sex chromosome conditions, you certainly ought to know about Turner and Klinefelter syndrome, and the autosomal conditions, Down, Patau and Edwards syndromes. The common microdeletion syndromes, Williams, Beckwith and Wheeler-Cardiofacial syndrome, also known as DeGeorge syndrome, commonly come up. And in the imprinted gene regions, Prader-Willi and Angelman, as well as Beckwith-Wiedemann sometimes, come up. In the single gene defects, of course, there's a huge range to choose from, but here are some of the commoner ones. The autosomal conditions, some of these you may have seen in the clinic. The recessive conditions, you'll almost certainly have met children with cystic fibrosis. And the X-linked conditions, Duchenne, Rett and Fragile X, are very common favourites with the examiners. And then, of course, it's important to remember that conditions which are often brought to the attention of geneticists may appear to be genetic, but the actual diagnosis is a non-genetic condition. But the important thing that, as the genetic specialist, one has to do is to distinguish between the genetic and non-genetic. And the commonest example of this is fetal alcohol syndrome. And so this hopefully gives you some indication of the kind of conditions you ought to be looking at. But of course I can't possibly suggest that this is a complete or total list that you should exclusively learn. So that's it for this lecture, and good luck with the exam.